Hello students, in the previous class we have discussed about the introduction and basic concept of biotechnology. In this class we will discuss about the tools of biotechnology, which is very important topic. This tool topic includes tools of biotechnology. It has some other subtopics: enzymes, cloning vectors, and then the host organism or host cell. You can just take it as host. After dealing with, we will come on the topic techniques. First, we are going to deal with the topic enzyme. Enzyme, it includes 5 types of enzymes. First, it is lyase enzyme. Second, it is restriction enzyme. Then third, it is phosphatase enzyme. Fourth, it is ligase enzyme. And fifth, it is phosphorylase enzyme. In this class, we will deal all these enzymes accordingly. So, let us see what is the lyase enzyme. Lyase enzyme. When we are going to separate or isolate the piece of DNA from any of the cell, either it can be plant cell, either it can be animal cell or fungus or any bacteria, we have to digest its whole cell wall, cell membrane or other nuclear membrane, so that we can just assess that DNA or nucleic material. If the cell is plant cell, if our cell is plant cell, then we need enzymes. First, as the cell wall is made up of cellulose, so we need cellulase enzyme. What it is? Cellulase. And the mature cell wall is also having pectin. So to digest pectin, we need the second enzyme, pectinase. Then the 2 more enzymes which is found in 2 more enzymes to digest the lipid material and protein material which found in the cell membrane. So it is lipase and the fourth is protease. These are the enzymes required to digest all the material, extra material, and to assess the nucleic material in the plant cell. Then if the cell is, our cell is animal cell. In animal cell, there is nothing like cell wall. There is only cell membrane, and this cell membrane is mainly having the lipids and protein. So we need only lipase and protease. Then if our cell is of fungus or fungi, as the cell wall of fungus is made up of chitin, So to digest the chitin, we need an enzyme chitinase. What it is? Chitinase. And along with this chitinase, we also need lipase and protease. Lipase and protease. If our cell is bacterial cell, if our cell is bacterial cell, to digest this bacterial cell, we need an enzyme which is known as lysozyme. Lysozyme. There is an additional information about lysozyme. In our body, there are some secretions like tears and sweat. It contains an enzyme lysozyme which digests the bacteria. So it acts as the it fight against any bacterial infection. So these are enzymes. Then the second enzyme is our restriction enzyme. It is very important enzyme. Second is a restriction enzyme. This restriction enzyme is also known as molecular scissor, as I have told earlier. Molecular scissor. Why? Because it has ability to cut DNA. It cut DNA at a specific site. Let us see. It is very important enzyme, or we can say the revolution related with biotechnology came just because of this restriction enzyme. So let us see what happened in 1963. It was observed that in E. coli, which is Escherichia coli, it is a bacteria having the two complex system as a defense mechanism. Okay, two complex system as defense mechanism. What were these? First, it was methylation, and second, it was restriction enzyme function. Why it was defense mechanism? These two systems were applied to reduce or to vanish any bacteriophage infection. Let us see what is this bacteriophage. Bacteriophage, these are those viruses which infect any bacteria. So these are viruses which infect any bacteria. Okay. So viruses which infect any bacteria. Let us see what happened actually. It is our E. coli. Okay. It is bacteriophage. It has inserted its nucleic material inside this. Okay. And in the E. coli, its genetic material is this Okay, having the same, as we know in the DNA there are the sequences or there is a nucleic basis. These are A, T, C, G. Okay, so there may be A, T, C, G, something like that. Or the same may be here also. Okay, so in the E. coli, the two systems, methylation and restriction enzyme. Restriction enzyme has the ability to cut DNA at specific sites. Let us see. Suppose this restriction enzyme, first it is restriction enzyme and second methylation. Restriction enzyme has ability to cut any, suppose any DNA piece between or at A and T. So it will cut this and also this. Okay, means along with this foreign DNA material, its DNA itself will be damaged. Means it will be just as a suicidal attack. So to protect this, there is another mechanism which is methylation. What happened in this case? In case of methylation, the DNA of E. coli itself is methylated. Here it is methylated, here it is methylated. So then the restriction enzyme will only recognize this piece of DNA and cut it. Means it will cut the DNA or foreign DNA of bacteriophage without disturbing its own. So it is like a defense mechanism because it is disturbing this one but protecting itself. So scientists has observed this capability of restriction enzyme and use this in further, and will discuss it later about the third enzyme was phosphatase. Phosphatase. I am saying ase. If we are using this suffix ase, means it is enzyme and it is working on this phosphate group. What is going to happen actually? It will remove the phosphate group. What is use of it? Let us see. It is your DNA. Okay. Circular DNA. Restriction enzyme had worked on it, this particular site. Okay. What will happen? This piece of DNA will open. This circular DNA will become linear. Here, one will be OH site and one will be phosphate group. As we know, the DNA material or backbone is just bind because of phosphatase bond. They will again join and they will again make this circular. This restriction enzyme will again work. It will become linear. They will again join. So to avoid this problem, as I have said, this restriction enzyme will work on this DNA which is circular. It will cut it in a specific site, and one side will be OH holding and the other side will be its phosphate group holding. But they can again join and form the phosphodiester bond, and again our work will be finished because it will become circular. To avoid this problem, we add phosphatase enzyme. Phosphatase enzyme. What will do this phosphatase enzyme? It will remove phosphate group. Phosphate group. So our DNA will become like this. Now it will remain linear. So with the help of this phosphatase enzyme, the DNA piece or cutted DNA piece, I should say, piece will remain linear so that we can work on it. Now our third enzyme. Now our fourth enzyme is ligase. Ligase. To create any recombinant, we have to add a foreign piece of DNA or our desired piece of DNA to the host DNA so that we can create a recombinant DNA. It will be like this. it is a plasmid, and it is a desired piece of DNA, so that we can just get our DNA, recombinant DNA. To get this, we have to join it. Okay, this ligase enzyme help in joining the DNA pieces. So help in joining. Then our last enzyme, which is our fourth enzyme, is fifth enzyme is phosphorylase. Fifth enzyme it is phospholipase. What happened with phosphatase? It was removing the phosphate group. Phosphorylase is just opposite to that phosphatase, because it adds phosphate group, which help in creating or forming phosphodiester bond, so that desired piece or isolated piece of DNA and plasmid DNA can be joined. Okay, and it will form recombinant DNA. Suppose it is like this. It is a isolated piece of DNA. It is plasmid DNA. Now the recombinant will be this. Clear? In helping this, it will be formed just because of this phosphodiester bond, and it will be done with the help of this phosphorylase enzyme. Okay, now let us see again some important topics or important discussion about restriction enzyme. Let us see some important points related with restriction enzyme. Restriction enzyme, it is mainly of two types: restriction exonuclease and restriction endonuclease. Exo means outside. What it does? It cut the DNA from end. Okay, means at the extreme. And what about endonuclease? Endo means inside. It will cut the DNA piece, cut the DNA in between, means like this. Okay, so restriction endonuclease, there are some scientific convention how to nominate them. What means? There is some nomenclature fundamentals for the restriction enzyme. It is like that: naming of restriction enzyme. It is very important one. Let us see. First, we mention a genetic name. Then specific name or a species name, then its strain, then the order in which we have found it, order of discovery. Okay, and the genetic name, it will be first later in uppercase. It will be first two letter in lowercase. First two letters in lowercase. Then a string. There are some issues which we'll discuss later, but normally it is first letter in uppercase. Order, it will be in Roman number. Let us see one example. There is a restriction enzyme E co R. Okay, E co RI. And how we are just getting this? Let us see. We are getting this restriction enzyme from Escherichia coli. Okay, so it is our genetic name, it is our species name. The first letter E is genetic name in uppercase. Coli, the first two letters CO from species name in lowercase. Then this strain is RY13. Which it is? RY13. So first letter in uppercase. And then it was identified first. Means this restriction enzyme from E. coli. E. coli is having many restriction enzymes, but the first restriction enzyme was this E. coli RY13 first. So we will mention this first. Okay. If we are getting some other restriction enzyme from this strain, we will just write like this. E. co R second. Okay. Now there is one more issue about we can use, if we had used this first letter of strain and it is common in other case also or other bacteria also, strain of other bacteria, then we will use second alphabet in lowercase. For example, there is some other case when the strain will be of same alphabet. Then what we will do? Then it will be not like that. It will be the second letter of strain in lowercase. For example, there is an enzyme HIND. This enzyme, this restriction enzyme, we found it from the Haemophilus influenzae. The strain was RD. So the H will come from here. Okay, will be in capital letter, genetic name capital letter. Okay, then first two letters of species. It is a species name, influenza, IN, HIN. Okay, then this R can be here, but already we had mentioned the R for E. coli or E. coli enzyme. So it will be D. So our enzyme will become the HIND, HIND. And this enzyme, there is an important point related with this HIND enzyme, that HIND enzyme second means the enzyme which we have discovered in order of second was first enzyme to be isolated. Very important it is. The first was discovered, but this was isolated first. It was about the nomenclature of restriction enzyme. Now there is important restriction enzyme is that restriction enzyme cut the DNA piece at particular region which is known as recognition site. recognition site. Recognition site is the region which will be identified by restriction enzyme, and it will be work on that piece of DNA. What is the specific nature of this restriction enzyme? It is always palindromic. Palindromic. What is the meaning of this palindromic? Normally, there is a palindromic means if we are reading some alphabet in the both direction, forward or backward, it will be same. So what exactly is palindromic? Generally, if any spelling is there and we are reading it from forward and from backward, it will be same. Means same from both direction. Okay. It is a general meaning of palindromic. In case of DNA piece, which is double stranded DNA segment, which is always double strand. There are two directions. One it is 5 to 3 and other it is 3 to 5. Okay. If we are reading from this side and from this side, it should be same for the palindromic. For example, what will be here? C, its complementary will be G, its complementary will be C, its complementary will be T, and here T, C, G. Just let us see. CGATCG. CGATCG. So it is a palindromic region. Okay. So palindromic, it is clear. This palindromic create a recognition site for the restriction enzyme which will recognize it and it will be work on it, means it will cut the DNA piece. Now this function of restriction enzyme, it is of two types. It can cut a DNA at a particular, means two ways. It is a piece of DNA 5 to 3, 3 to 5, A, G, C, then we will just get T, C, G. Here it will be T, C, G. It is a palindromic from 5 to 3, A, G, C, G, C, T. From here, A, G, C, G, C, T. Okay. So means from both sides we are getting the same sequence. Now, if the recognition site or if the site where the restriction enzyme works is this and this, means A for both strands, it will cut it in this form A, and it will be this region. It will be like this, G, C, T. Okay, and the second, but we are just getting A and this sequence, T, C, G, C, G. Here it will be joined like this. Okay, double hydrogen bond. Here it will be triple hydrogen bond. This G and C always joined by the triple hydrogen bond, and T and A always joined by the double hydrogen bonds. Okay, so after this type of cutting, we are getting the overhanging portion of the DNA string. These are known as our sticky ends. Sticky ends. Why they are known as sticky ends? Means when they come closer, they can again join because they are just complementary with each other. And this sticky ends DNA segments are very useful in the creating recombinant DNA technology. It was the first way. And second way, if it recognizes the C, if it recognizes C, means it will cut it here and here. Then we will get our DNA segment AGC, here TCG. The same will be TCG AGC. Double hydrogen bond, triple hydrogen bond, triple hydrogen bond, CNG. Hydrogen bond will be always represented by dotted areas. Okay. Then in this piece of DNA segment, there is not any overhanging portion, and this is known as a blunt end. Blunt end DNA segment, and it is not very useful in case of biotechnology. So it was all about the restriction enzyme, how it works, what is the nomenclature of restriction enzyme. Let us see once again what we have done. In short, we have discussed about the tools of biotechnology, and its subtopic was enzyme, and so it was all about the enzymes associated with the biotechnology. Thank you very much.